Greetings, Mr. Del Prado's science students. Welcome to Mr. Del's review on biochemistry. Please remember that there is going to be a test tomorrow, which is going to be Thursday. I know that I'm not here today, but I'm here digitally, and I'm going to try to help you guys review for the biochemistry test, which is going to be tomorrow. So if you look, right, we're doing biochemistry. We'll start off with a little periodic table, and we'll talk about water, talk about pH, and we'll talk about the four major molecules that are in your body. I promise to try to go through this as quickly as I can so that you're not bored by hearing my voice while I'm not even in class. Okay, so I think you should know what an atom is. I think you should know that there are protons, neutrons, and electrons in atoms. I think I'll probably ask you maybe about like the positively charged subatomic particle, that's a proton. The negatively charged subatomic particle, it's an electron. The neutrally charged subatomic particle, it's a neutron. Protons and neutrons are in the nucleus. Electrons are flying around the outside. Okay, there's a picture of some protons and neutrons, and you can see the electrons, they're flying around in those energy levels outside the nucleus of the atom. Okay, there's the charges, and there's the mass of protons, neutrons, and electrons. The nucleus, remember, is going to have your protons and neutrons inside of it. The electrons will be flying around the outside. Okay, what I'm going to ask you to know about the electrons is that they exist in these energy levels outside the nucleus. The first energy level is filled and most stable with 2 electrons. Each energy level after that will be most stable if the atom has 8 electrons in its outer shell. Remember that atoms will beg, borrow, or steal in order to get 8 electrons in their outer shell. That's called the octet rule, and we'll talk about that in just a bit. Okay, so there's our elements. I'm going to give you a periodic table on for the test on Thursday. Remember that there's a couple things you can do to make your life easier on the periodic table. If you can put the number of valence electrons and charges at the top of each column. Remember, we did that in class. I would encourage you to do that when you get the periodic table on Thursday on your exam. Remember, you should know the difference between an ion and an isotope. Ions are atoms that have gained or lost electrons. Isotopes are atoms that have the same number of protons but different numbers of neutrons. Okay, another look at some of the numbers from the periodic table. You can see the atomic number then for hydrogen would be 1. The atomic mass, remember, is an average of the different atomic masses for hydrogen, and then you get that 1.007 AMUs. Isotopes we talked about. These are atoms with differing numbers of neutrons, so it affects their atomic mass. Molecules are, these are things that combine when 2 of the same atoms actually form a bond with one another. Compounds are when 2 different atoms combine with one another. Example of a molecule would be O2, that's oxygen bonded to oxygen. Example of a compound would be NaCl, that's sodium bonded to a chlorine. You should know essentially the molecular formulas, but look at that big one down there, C6H12O6, that's glucose. I would expect that you would know that that's the molecular formula for glucose, and I suspect most of you know the molecular formula for water, which is going to be H2O. Okay. The octet rule is that atoms will share electrons or they will steal electrons from one another in order to get their outer shell filled. For the most part, most atoms are most stable when they have 8 electrons in their outer shell. If an atom gains an electron to have its outer shell filled, it will become a negative ion, also known as an anion. If an atom loses an electron, it's going to become positively charged. That's called a cation. Okay, water is one of the classic molecules in which we have a covalent molecule. This is where the atoms are actually sharing electrons. The hydrogen has 2 in its outer shell, so it's stable. The oxygen has 8 in its outer shell, so it is stable. Water then is a covalently bonded molecule. Ionic bonds occur when one atom steals an electron from another atom. In this case, you can see chlorine stealing an electron from sodium so that both of their outer shells end up having 8 electrons. The ion we talked about, when you gain an electron, you become negatively charged. That's an anion. When you lose an electron, you become positively charged. That's a cation. We've already talked about molecules in here, and I think we've already talked about covalent bonds in here as well. Okay? So that's it essentially with the periodic table. I think you should be able to tell me the number of protons, neutrons, and electrons for each atom on the periodic table. You should be able to tell me the number of valence electrons, and you should be able to tell me the type of ion that it would form. Remember, the valence electrons and ions you would be able to get by using that the cheat sheet that we did in class on the top of each column. That brings us then to the ultimate molecule that we talk about in biology, and that is water. Water is a very unique molecule because it is a polar molecule. It has a positive end and a negative end. I think you should know that the positive end of water is the hydrogens, the negative end is going to be the oxygen. And that creates then an attraction between water molecules. Remember that opposites attract. So you have the positively charged hydrogen attracted to the negatively charged oxygen of a different water molecule. That is called a hydrogen bond. Remember, hydrogen bond isn't really the correct name because it's more of an attraction than it is an actual bond. But it is the attraction between the positively charged hydrogen and the negatively charged oxygen of water, which is a polar molecule. You can see the water molecules there, kind of like bumping back and forth from one another. Again, usually you get the negatively charged oxygen attracted to the positively charged hydrogen. This hydrogen bonding in water gives it some unique properties and that allow for life to continue here on Earth. One of the things that the hydrogen bonding does is that when water freezes, the hydrogen bonds kind of push out and it pushes the water molecules away from one another. That makes ice, which is solid water, that makes ice less dense than water, and that's why ice floats on water. The other thing that these hydrogen bonding allows is it allows water to stay attracted to itself. That's called cohesion. You can think about the drops of water on the penny and the dome that those drops of water made on the penny. Then you can think about adhesion. That's the ability of water to stick to something else that's not water. You could think about a plastic water bottle and the water droplets that like form on the side of the water bottle. That's an example of adhesion. Okay, moving on from water then, remember that water is one of the most important molecules here on Earth. And in water you have H2O. And those hydrogens and those hydroxides can lead to molecules called acids or bases, which are kind of important in living things. Okay, so acids are solutions that have lots of hydrogen ions in water. Bases are solutions that have lots of hydroxide ions in water. Hydrogen ions are H plus ions. Hydroxide ions are OH minus. Remember too that sometimes in an acid the hydrogen will bond to water and it will form an H3O plus. That is called a hydronium ion. Acids and bases are measured using the pH scale. pH of 7 is neutral. That means there's an equal amount of OHs, which are hydroxides, and H's, which are hydrogen ions. Water is a classic example of having a pH of 7. It is neutral. A pH below 7 is considered an acid. It has more hydrogen ions in solution. A pH above 7 is a base. It has more hydroxide or OH molecules in solution. That then brings us to the biggest part of our biochemistry unit, and that is the macromolecules, or the 4 big organic molecules that are found inside living things. The first one is a carbohydrate. Remember that carbohydrates are going to be used for energy inside of our bodies. Okay, the basic ratio for carbohydrates is that it's going to be a 1 to 2 to 1 ratio of hydrogen to oxygen to carbon. And if that's confusing to you, that's okay. Just think about the ultimate carbohydrate. That's glucose, which is C6H12O6. 6 carbons, 12 hydrogens, 6 oxygens, a 1 to 2 to 1 ratio. The building block of carbohydrates are monosaccharides. That is a single sugar unit. They can sometimes connect together to form disaccharides. That's 2 rings, or polysaccharides. That's 3 or more rings. The ultimate polysaccharides would be things like glycogen, which animals use for energy, and starch and cellulose, which plants use for structure and for energy. There's an example of a monosaccharide. You can see a disaccharide as well, and then the polysaccharide on the bottom right corner. Those are all examples of carbohydrates. Lipids are another organic molecule. They are also used for energy. These things are real long chains of carbons and hydrogens. Both lipids and carbohydrates both have carbon, hydrogen, and oxygen. Lipids are long chain molecules that are used for long-term energy sources. Okay, the basic building block of a lipid is a fatty acid. Fatty acids can be either saturated or unsaturated. Saturated fats are solids at room temperature. They have all single bonds and they tend to be straight molecules. Unsaturated fats tend to be liquids at room temperature. They have carbon-carbon double bonds, and you can see that at the bottom of your diagram there. And they tend to be bent, which makes them easier to break down. But lipids and carbs both have the same function inside living things. It's just carbs are used for quick energy, lipids are used for long-term energy. Phospholipid's a special type of lipid that's found surrounding your cells. I'm glazing over that because we spend a whole unit talking about your cell membrane. Okay, steroids we talked about in class. These are specialized types of lipids that can tell your cell to do different things. They are actually found in the blood and they attach to receptors on your cell membrane. Proteins are a big one. Proteins do have multiple roles inside the body. They can be involved in structure, support, and movement, communication. They can transport molecules. They can also, like enzymes, carry out chemical reactions. Proteins are made up of amino acids. That is the monomer or the building block of a protein. Amino acids are connected together by a special type of covalent bond called a peptide bond. Proteins, you can see some amino acids there. You can see that R group in the top right-hand corner of that one amino acid. Remember that there are 20 different amino acids on Earth. It is the arrangement of these 20 different amino acids that make us different from one another. So again, of all the living things on Earth, there's only 20 different amino acids that make up all the living things on Earth. Okay, amino acids can take up different shapes based upon how they fold. The first type of folding we see in amino acids is called the primary structure. That's just the sequence of amino acids that are running from one to the next. These amino acids can then fold on each other like sheets of paper. That's called a pleated structure or a beta pleated sheet. Sometimes they can twist on each other like a coil. That is called an alpha helix. If you look down at letter C, these alpha helices and these beta pleated sheets can then fold on each other to make these ribbons. These are tertiary structures. And then finally, you can get multiple tertiary structures that kind of get attracted to one another, and that will make up a single protein, and that is the quaternary structure. Here's a picture of some collagen on your right-hand side. There's some hemoglobin on the left-hand side. And here's a great look at some alpha helices in the hemoglobin protein, which remember is found in your blood and carries oxygen around your blood. Before we get to nucleic acids, I'm just going to remind you there are some big-time proteins out there that I think you should know. I think you should know collagen. That's an important structural protein. You should know keratin, makes up your fingernails and your hair. Hemoglobin's a good one, and there's a protein hormone I think you should know, and that is insulin. It helps glucose get into your cells. Last big molecule in our bodies are nucleic acids. I don't spend a lot of time on this because DNA is the ultimate nucleic acid. We're going to talk about that later this year. Nucleic acids, the monomer for a nucleic acid is a nucleotide. Nucleic acids have carbon, hydrogen, and oxygen. They also have nitrogen, but the big thing that they also have is phosphorus. And that's what separates out a nucleotide from an amino acid. Remember, amino acids have carbon, hydrogen, oxygen, and nitrogen. The nucleic acid has all those things and a phosphorus as well. Okay, DNA and RNA, they give us our genetic material. These are the traits that get passed down from our parents to us. Okay, the basic part of a nucleotide is a sugar, a phosphate, and the base. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. I'd like you to know those names, but we'll get more into those when we actually do our DNA unit. You're not going to need to worry about chemical reactions. We'll talk more about reactants and products at another time. I hope this review was helpful. I hope you were able to answer some of the questions that go with it. Feel free to email me tonight if you guys have any questions on the test itself. Thanks a lot, and hopefully I'm going to see you on Thursday.